Hi there. Today I'd like to share a procedure by means of which you'll be able to clean a protein. It's a Pymol. I have already uploaded my desired protein in Pymol. And after uploading, actually how to upload a protein in a Pymol. First of all, I can select my desired protein. And when you will upload this protein, then obviously it will be visible on Pymol. But first of all, you can download this specific protein from PDB, that means RCSB PDB file, and I will also attach this specific link in the description portion. It's a 4J6CU. It's a protein ID. I have downloaded this protein from PDB, that means Protein Data Bank websites, and then I will upload this specific protein in Pymol. I will press the right button and then open with Pymol Molecular Graphics System. When you will press Pymol Molecular Graphics System, then automatically after a while your desired protein will be visible. Then when the uploading session is done, then you have to select a specific portion that is called SEQ, that means that indicates sequence of this protein. I will drag to the bottom and I will press SEQ. You can say it's a sequence. When you will press this SEQ, that means sequence, then obviously the overall sequence of this protein will be visible in the upper portions of this interface. And then I will delete the heteroatom and outer molecules. So the red portion circle or zero that indicates the outer molecule, I'll delete this specific portions. I'll press here and then I'll press shift and I'll drag, and then I'll select this portion, and automatically all red colored uh, O will be selected, and I'll press right button and press select remove, and automatically outer molecule will be removed. And the next one, I'll select the remaining portions of red colored O, and I'll press shift and I'll press again this last red colored O, and then I'll select right button of my mouse and then I'll press remove. Then automatically outer molecule from this chain will be removed, and in this way you'll be able to clean your desired protein. And if you'd like to rotate this specific protein, then you'll be see that there is no any heteroatom and outer molecule. That means this protein molecule uh, already cleaned, or you can say had been cleaned right now. And if you'd like to save, then you can also say save from the file menu. And if you'd like to select get PDB, then automatically in PDB format your overall clean protein will be downloaded. And if you'd like to select export structure, then you can also select export structure, export molecule, and then you can download your clean protein. I'll select praise save, and then I'll select PDB, that means it's a protein data bank, that is the safest type. And if you'd like to select if you'd like to give a specific name, you can give a name, it's a clean protein. I'll select my desired folder and then I'll find out my desired folder again. It's a protein I have already cleaned before starting this video recording, and I can select it's a clean to protein, and I'll press save. Then automatically our clean protein will be saved. In this way we'll be able to clean our desired protein. The another way I would like to share with you, you can easily clean your desired protein. how can you clean our desired protein in different way? First of all, I will select unclean protein. I will press right button of this specific structure, and then I will open with Biovia Discovery Studio Visualizer 2024, this software. Then after a while, you'll be able to see 4J6U. It's an unclean protein will be visible to your Biovia Discovery Studio Visualizer software. And then when you'll be able to see uh, your desired protein will be visible to this specific software. You are seeing that the overall structure, that means 4J6U is loading. And when the uploading session will be done, then I will remove our unclean protein. I will remove different kinds of heteroatom and outer molecules from this unclean protein. You are seeing right now our 4J6U protein is visible right now on the interface of DiscoveryStudio Visualizer, and then I'll press this specific arrow button, and I'll select. When I will select this specific button, then overall structure will be invisible. I'll select here. I will remove outer molecule. That's why I'll select outer, and I'll select right button of this specific text, and I'll select remove group. When I'll select remove group, then after a while you'll be able to see outer molecule will be disappeared, because I'm trying to remove outer molecule from this overall structure of protein molecule. So next on, when I'd like to select heteroatom, I'll select and right button of my mouse, and I'll select remove group. Then automatically heteroatom will be removed from the structure of our protein molecule. In this way, we have removed heteroatom and outer molecule from our desired or target protein, or you can say desired or target unclean protein. And the next one, if you'd like to select B, I would also remove the B chain. I'll select right button of this mouse, and I'll select card. You can also card. When you'd like to select card, then automatically the uh, your selected chain or selected group will be disappeared or removed from your structure. Here is another portion. I'll also remove this add chain. I'll select right button of this mouse of my mouse, and then I'll select card again. Then automatically the you can say desired B chain will be disappeared. And if you'd like to see the overall clean protein, then you can easily see. I will remove, I will rotate this clean protein. Actually, when you'd like to rotate this clean protein, then you'll be able to see there is no any existence of outer molecule or heteroatom in this protein structure. That means our protein is clean totally. So guys, this is all about today. If you'd like to clean your protein that you have already downloaded from RCSB PDB website, then you can easily clean your desired protein from or you can say using Discovery Studio Visualizer, at the same time Pymol software. And the next one, if you'd like to save this structure, then I'll select file, I'll select save. Again, I can select Discovery Studio Visualizer save as type from. I'll change protein data bank files, that means PDB files, and I'll change the file name. I'll change the file name. Clean. It's a clean using Discovery Studio Visualizer, DSP, Visualizer. And the next one I'll press here. Actually, the cleaning portion is done right now for our docking, for molecular docking. We know that about our protein must be cleaned, and the next one, our you can say protein must be you can say energy minimized. The energy minimization process will be done by means of SwissPDB Viewer. Again, I'll select SwissPDB Viewer from my desired folder. Obviously, I will select right button, and then I'll select research softwares and SwissPDB Viewer, that is SPDB. It's a SwissPDB Viewer. I'll select you can say SPDB, that means Swiss-PDB Viewer applications. I'll press double click and I'll press cross of the upper window, and then I will select file. I will select open PDB file that I have already cleaned. I'll select any kind of you can say clean protein. I'll select 4GCU clean that I have already downloaded from SwissPDB Viewer. I'll select here and then I'll select press open, and then automatically you'll be able to see your another window will be visible. If you'd like to energy minimize, then you can select file, you can select portion, select menu, and then select all. That means I have already selected the overall protein, and I'll select bill, and add add hydrogen. That means I have already added hydrogens with a B to added any kind of lost hydrogens. And the next terms that is called, you can say, tools. When I would like to use this menu, I'll select tools and I'll select energy minimization portion. When I'll select energy minimization, then automatically you'll be able to see the energy minimization procedure will be started. Then you are seeing that the energy minimization process is right now ongoing. That means it's progressing right now. And when this specific black slider will be in the end portions, or you can say at the right end portions of this box, then you can say that uh, our energy minimization process has successfully done or has successfully completed. In this way, we can prepare a protein for molecular docking. First of all, we have done cleaning by means of using Pymol, at the same time by means of using Di- Discovery Studio Visualizer. And the next one, we have uploaded this specific clean protein on SwissPDB Viewer. And right now, the overall you can say the overall energy minimization process is completed. After energy minimization completed, you will be able to see the overall energy of this protein structure. Overall energy is, you are seeing that on the doc portions, or you can say text portions, energy is minus 34,735.03 on kilojoule per mole. This is the overall energy of this protein structure after energy minimization procedure. I can cross this specific window. I'll press file. I'll press, uh, you can say, save portion. I'll press save. When I'll select press without pressing, then you'll be able to see current layer will be visible in the another window. I'll press current layer, and then I'll select save as type will be PDB files, and I'll select this protein. It's a, you can say, protein that I have already cleaned, and I have already energy minimized. That means I'll use energy minimized minimized. That means uh, cleaning and energy minimization is the two procedure to prepare a protein for molecular docking that I have already done by means of using two software. Number one, you can say I can clean a specific protein structure by means of Pymol software, and the next one you can say Biovia Discovery Studio Visualizer, and the next one I have to energy minimized. I have to minimize the energy of the overall structure of protein by means of SwissPDB Viewer 4.1.0 version. And when the cleaning and the energy minimization procedure is done, successfully done, then we can say that our protein is completely prepared for our molecular docking procedure. So guys, this all about today. If you'd like to know about any kind of information that is related to molecular docking or how to prepare a protein, then you can share your opinions, share your questions or queries in the comment box. This is all about today. Thank you for watching this video. Stay tuned, stay connected with this channel. Hit the subscribe button and press the bell icon for watching this kind of videos again and again or in the future. Thank you guys. I'll remove this kind of interface because it's not the essential right now. I'll press cross and again I'll press cross here.